Guys, welcome to your first biology video at Williams Secondary Montessori. First, we're going to be talking about the nature of biology. Specifically, we're going to be looking at experimental design today. This should be pretty familiar for most of you, as you saw it all throughout middle school, and some of you in ninth grade. But we're going to be reviewing this and then discussing it further in class. How do you design an experiment? Okay, that's the big question. It's one of the key pieces that helps us define science. Okay, any video that is going to be posted in any of these PowerPoints will be posted to supplement them online. There's a video here about autism, which is a very testy subject in the field of science. So the first part of designing an experiment is finding the problem and the purpose, or the purpose, depending on what type of experiment. So what is the problem? What are we trying to define? What are we trying to do? Okay, this is the question that the lab will answer, and it has to identify the independent and dependent variables. Okay, the independent variable is the factor the scientist changes from group to group. So say we're having an experiment where we're testing fertilizer, types of fertilizer. The independent variable, which is what we're changing, would be the type of fertilizer that we are using, or if we're not using fertilizer. Okay, the dependent variable is what we're going to measure. So if we're testing fertilizer types, what are we going to measure? We're going to measure, say, height of the plant. So the dependent variable, what we're measuring, is the height of the plant. Okay, so there's two more videos here that you will find links to on the class website. So furthermore, we have to, even before we do an experiment, we have to do background research. Any scientist is going to go, well, no longer into libraries, but more online, and start looking to what research exists. We don't want to be repeating what already exists there. We want to be furthering the study of science. Okay, so we have to learn about the topic. You're never going to do research on something if you don't know anything about what you're doing. Okay, this background research helps us come up with an educated hypothesis. Okay, you always hear hypothesis is an educated guess. What does that mean? That means I've done research so I can make a reasonable hypothesis, a reasonable guess at what's going to happen. Okay, we're not just shooting in the dark all the time. Okay, so what is a hypothesis? Like I said, it's an educated prediction or an educated guess. Okay, it has to be testable. You have to be able to prove it false, and it needs to agree with the previous research. You can't be going out in the dark and being completely contrary if all the research says one thing. Now, if all research says one thing, you have a hypothesis that's supported by the research, and then you find against it, you prove your hypothesis false. That's fine, but you have to support it with the data. Okay, so then we have our procedure and slash experiment. Okay, your steps, they must be precise and detailed. Okay, when you write up a lab or you write it out for publication, it has to be detailed enough that somebody can repeat your experiment, and we'll get into why they have to be able to repeat your experiment. Okay, you must have what's called a control group. Okay, this is normal conditions. So we were talking about working with fertilizer. Your control group would be growing your plant without fertilizer. That's normal conditions, or the absence of the independent variable, and it is used as a point of comparison. Okay, you can't say this fertilizer works better if you don't have a control group. Okay, all right, here's another really important thing. Experimental groups only have one changing variable. You can only change one thing at a time. So many of you know that I have a new baby at home. She was born last January. When we're trying to figure out why she's crying, we change one thing at a time. Okay, is it that she needs a diaper change? So we change her diaper. Okay, is she still crying? Okay, is it because she needs food? Check for, feed her food, see if that stops her. Or is she just being contrary? I mean, you aren't going to do three things all at once, change your diaper, feed her, and do all this and try to be able to isolate what was wrong. Okay, then we have observation data. Okay, they have to be objective. Okay, they can't be subjective. What does that mean? Okay, so a good example is the bacterial colony is yellow. You're describing something that everybody else can see. Okay, except for maybe the colorblind, but that's another issue. Okay, a bad example, the bacterial colony is nasty. Okay, some of you will think the bacterial colony is nasty. There are those scientists who find bacterial colonies awesome. Okay, it's just like how most people think cloth diapering is crazy, but I think it's absolutely fabulous. Okay, that would be subjective, but color versus opinion. Okay, you need to go for objective. Okay, you need to use measurements, okay, whenever possible, okay? You're not trying to insert your own opinion, so measurements are free from opinion, okay? Another example, 50 bacterial colonies. Everybody can count 50 bacterial colonies, okay? There are a whole bunch of colonies. What defines a whole bunch? Okay, it's like that. What describes a pair? What describes a few? What describes many? A whole bunch? That's very subjective. Okay, so measurements are great. Numbers are fabulous. All right, furthermore on observations and data. Okay, in our observations and data section, we're not going to draw conclusions, okay? You are just looking at the data, all right? We're not saying this is true, this is not true. So an example, there are bacterial and fungal colonies in the shoe section, okay? Bad, the shoe was really dirty, so it was the most contaminating and grew a lot of stuff. There, you're assuming that the shoe was the most dirty. You're drawing a conclusion. Okay, you're not stating simply what the data shows. All right, and it should be very organized. Organization is key. We're in Montessori, you should know organization. Scientific data, all about organization. Okay, we love graphs and tables and drawings of apparatuses. So if you're setting something up complicated, okay, you would want to have a drawing. So for example, when I worked in research, we used what was called a T-maze to test the working memory of our mice. And we would often include, though a T-maze was pretty common, you could include a picture of what your T-maze looked like in that apparatus so people can understand. It's a maze that's shaped like a T, but a drawing is very helpful. Okay, and you always must use units of measurements. You have 3 whats. Is it 3 inches, 3 meters, 3 colonies? You need to have that unit or the number means nothing. Okay, and everything should have a title. Tables have titles, charts have titles, graphs have titles. Otherwise, I don't know what I'm looking at. Okay, that tells me what I am doing. All right. Then we move on to the analysis and the conclusions. So here you're going to answer your purpose question. So answer your questions. Okay, you're going to accept or reject your hypothesis. Were you right in what you determined? Does it go contrary to all the research that you've seen? That is very possible. Okay, and then you can explain what you see in the data. Okay, I see 50 colonies when I swab the shoe, and I only see 20 colonies when I swab the sock. Okay, whatever you see. Okay, another video that you will find linked on the website. All right, so how do we know it is good science? There's a lot more bad science out there than there is good science, and so many of the articles that you'll read on mainstream websites, your CNN, your, USA Today, whatever you're looking at, the Skim, right? Or link to Facebook, okay? That's often bad science. Good science, okay, must be shared. We share our work with other scientists, and then those scientists repeat and verify the work. So they say, yes, what they have done is right. It's called peer review. And actually what you do when you submit any work to a peer-reviewed journal is you have to include a list of people in your field who are qualified to work the work that you've done. Okay? You don't and then the people on the board for that journal will pick from those people and possibly somebody they want, but you give them suggestions of people who are working in the same field who know what you're doing. So they're not just so you don't have a person who works on prostate cancer trying to do something with brain cancer because that's not their specialty. So it's people who are specialized evaluating other specialized people's work. And this is called peer review. All right. And all scientific knowledge is tentative. Okay. It can always be changed. Okay. Theories are the most logical explanation based on current evidence. And they become stronger every time more evidence that supports that idea is gathered. Okay. Laws. Okay. When it's a law, it's a big deal. Okay. That's universal generalizations that are virtually unchanging. Okay. So we have the law of gravity. Here on Earth, if I take something and I drop it, it's going to accelerate 9.8 meters per second towards the floor. Okay. Theories. Theory of evolution. There is significant evidence that supports that theory. And as we continue to gain evidence that makes that theory stronger, okay? But it's not quite to the point of being a law. So we have two works here that you're going to be writing down, okay? I'm going to show them to you, and then your job will be to go back, make sure you have answered the questions to present them to me in class, okay? So Scientific Methods series, part 1B, all right? So Dre collected some bugs and he separated them into 3 groups, right? He has this data. He has 3 groups, A, B, and C. He gives them different hours of light per day, and then he records the days lived. You need to write down the control group, the experimental groups, the independent variable, the dependent variable, and give a reasonable hypothesis, okay? All right, I'm going to move on, but remember you can always come back and pause the video at this point to complete any questions you haven't finished. Here's another one. All right, we have Barnum received 12 labels in the mail. He put 4 on plastic, 4 on wood, and 4 on paper. All right, the normal place for a label. Okay, he recorded the number of days the labels remained attached. Once again, please write the control group, the experimental groups, the independent variable, the dependent variable, and the hypothesis. You can pause on this slide as long as you need. All right, this is the end of your first video on experimental design. Make sure you have your notes and the answers to the 2 questions that have been presented to you brought to class the next time I see you. Have a great day.